Hi, Amanda. Thanks so much for joining me on this episode of Concentrating on
Yeah, thank
Chromatography.
you so much, David, for the invite.
To start, can you tell us a little bit about your background and your journey in analytical chemistry and what first drew you to chromatography and mass
Well,
spec?
I was introduced to science during my graduation in pharmacy biochemistry, and I felt in love with analytical chemistry. So, what drove me then to start an internship in the analytical chemistry lab at university, working with chromatography applied to botanical medicinal products analysis. So, later, I started my PhD in analytical chemistry, working with LCMS based metabolomics, which opened actually the door to learning more about design of experiments, driving me to apply it later, supporting the development of methods used to ensure the quality of medicines. But in fact, what really totally got me into chromatography and mass spec was the exciting work I have done during the time I worked at the United States Pharmacopoeia Reference Standard Lab. So where I used, these techniques, chromatography, mass spec, day in and day out to characterize different standards, with a major focus on small molecules, drug substances, chemical medicines, dietary supplements, botanical ingredients, expanding later then to peptides and proteins, and also like applying design of experiment and modeling to understand further chromatographic separation mechanisms.
Yeah, I was very impressed looking at your background in terms of you've just worked across so many different areas, like you mentioned, the small molecules, but also peptides, proteins, and now biopharmaceuticals. How did your career evolve across all those areas? Were you pivoting to subject matter you were more interested in, or did the opportunities just come to you that
I
way?
think I was too curious. I wanted to expand knowledge. So during my time at the US Pharmacopoeia and throughout my PhD project, in fact, I primarily worked with the small molecules. And this experience gave me a very solid foundation in method development and in structural elucidation of compounds. So I had the opportunity to work extensively with high resolution mass spec approaches, exploring different kind of fragmentation mechanisms such as CID, HCD, UV photodissociation, all that. And structural elucidation was, in fact, one of my passions, sincerely. And I wanted really to expand my knowledge to new types of products at USP. Yeah. And I was too curious then, and I started volunteering myself to become visiting scientist at some universities across the globe. Like one example, I worked proteomics at the Technical University of Berlin. And the main goal was to acquire LCMS and MS data to identify proteins and peptides in target samples. And I was also excited to work with Orbitrap for the first time at that time. And then later during my PhD, I started working with Orbitrap, doing MS analysis, and also working at the Leiden University, doing now method development using high resolution mass spec for bioanalysis, like analyzing liver samples and all that, trying to extract bioacids from liver samples, plasma, and all that. And then later at USP, I shifted then slowly towards trace impurities analysis, like using low and high resolution MS. This was mainly driven by a hot topic, probably you have heard about that, which is nitrosamines. Yeah, so I was developing methods to monitor those impurities, which are considered as genotoxic impurities. And we also monitor them at very low level, and I started teaching mass spec to regulatory agencies, which was like really interesting for me, because I love teaching, and I was teaching this topic and also how to validate LCMS and GCMS methods to regulatory agencies. And after I left USP, I started working at a contracted research organization called RIC Group. They are based in Belgium, and the main topic became then really large proteins, peptides, now working also with oligonucleotides, RNA therapeutics. And these are quite exciting products. I could learn even more about chromatography, different aspects that impact method performance, and deepen my knowledge on separation mechanisms.
The primary audience of this podcast is
Oh,
undergraduate
okay.
chemists. So they might be very interested to hear from you. What did the day-to-day work look like as a chromatographer?
Okay, that's a challenging question. Actually, we learn so much throughout our careers, but I think I can summarize it in one sentence. I would say, like designing experiments, troubleshooting methods, and interpreting unexpected results. I would say I always dedicated significant time on doing data analysis and like taking the data and seeing what the data is telling me so that I can take a good decision, optimize methods, and so on, and also learn about chromatography. Yeah.
Well, I think that's a great answer. That's very insightful. Many of our listeners hear the term analytical quality by design, but they may not fully understand it. How would you explain that concept in simple
Yeah.
terms?
So, analytical quality by design, or AQbD in short, is essentially a systematic approach to understanding how analytical methods they perform. Yeah. So it relies on tools like risk assessment tools to help identify what factors could influence the method and where variability can come from. So, in simple terms, it would help us move from just running a method to really understand what impacts its performance and why. So we can design conditions that are more than robust and better optimized. There are several of risk assessment tools that can support this approach, the AQBD approach, I mean. For instance, design of experiment is one, a very powerful one, because it allows us to study multiple variables at the same time and understand also their interactions. So, in short, AQBD approach reflects the essence of quality by design. Yeah, so we build quality into the method from the beginning, based on foundations of quality by design, which is quality risk management, rather than trying just to fix issues later during routine use. We want to build the quality during the beginning of the product, the not the product, but the procedure life cycle. So then the result is basically greater confidence in the method. We can reach like less variability, perhaps fewer out of specification results, more reliable decision making. And it's interesting for the undergraduate students, but also scientists from industry perhaps also to know that these concepts they gained a strong momentum with the IC8Q8 guideline published in 2000 and beginning of 2000 on pharmaceutical development. So those guidelines they set a stage for the industry in terms how to propose new molecules to regulatory agencies, etc. So they're quite impactful for the pharmaceutical industry. And the quality by design principles gained momentum exactly with this first guideline in 2004, IC8Q8. And more recently, the IC8Q14, this is another guideline which discusses now enhanced approaches for procedure development based on analytical quality by design principles. So I just wanted to mention this because, as you said, so the audience is basically undergraduate students, and sometimes you're thinking, okay, where should I go? Where can I go with chromatography, right? So, and method development. So it's nice to know about some of those, regulatory guidances, which is driving actually how the industry is working and also showing where we need skilled scientists, working and knowing about all those topics.
One of the reasons I was really looking forward to speaking with you, Amanda, was because I see this concept as really interesting and important. And this is the first time we're discussing it on the podcast. And I think it's probably somewhat straightforward how this differs from the traditional trial and error approach. But I'd love to hear any details you have on that, in addition to what some of the biggest benefits might be of using this approach.
So, what is the traditional approach? We can call this trial and error approach, right? We can call this also as one factor at a time approach because we change one factor and we see how this is impacting on the response that we are studying, right? And we will have some limitations in terms of getting knowledge on how those factors are impacting on the performance. So the analytical quality by design will be a systematic approach where you can select suitable experiments to run and you can do multivariate exploration of the parameters. So you can test them, so you can vary them all at the same time. And this is the biggest difference between the one factor at a time and the systematic multivariate experiments that usually we do when we implement AQbD. So with that, we can reach more knowledge. So we can define not just a final method or a final process. So you can define actually operable ranges. Yeah. You can have more predictability. You can predict good performance and where you have bad performance, even if you didn't test. We can even work with modeling, for instance. Yeah. And when you compare to the one factor at a time approach, we have some limitations because we cannot characterize, for instance, interactions between the different factors that might be, significant. So we don't know if we vary the factor 1 and 2, what is actually causing a change on the response. So we can then get more knowledge with the AQBD approach.
No, that makes a lot of sense to me. In terms of the labs who adopt it, what would you expect to see as in terms of the largest benefits for moving to this approach?
Well, I think one of the biggest benefits would be using DOE and modeling so that you can achieve much more knowledge with fewer experiments. Like instead of testing one factor at a time, you can explore multiple variables simultaneously. And this is especially powerful because it allows us to identify interaction between variables that can be significant. For example, in chromatography, let's take an example, otherwise it's too philosophical. So, how in chromatography the pH of the mobile phase and the gradient slope that you designed might work together, impacting on performance. Yeah. So these significant interactions, they're often completely missed, in traditional approaches. So you cannot see that this interaction is impacting on performance. So you select a final method, chromatographic method, and in fact, this might not be so robust. Yeah. So it also enables us to do predictions. Yeah. Now we can predict responses even if you didn't test, and this is amazing. So you don't need to spend a lot of time in the lab doing several experiments without knowing when you will find the optimum. And so this is expanding knowledge then. Yeah.
Now, one thing that I think would be helpful to explain is design of experiments the same as ABQD, or are they different?
Yeah, so this is a very nice question because often we see like scientists, especially, I will not lie, especially in the pharmaceutical industry where the discussion of AQBD is pretty strong right now. So they really think, okay, I will learn DOE and I know how to implement AQBD. Yeah. So while DOE is very powerful tool, so using DOE alone does not mean we are implementing the analytical quality by design principles. So AQBD is a broader mindset that includes risk management and also deep understanding of method performance. So tools like DOE will help us map good and bad performance regions, which will strengthen then risk assessment and help identify the optimal conditions. But identifying risks is only part of the story. We also would like to define counter strategies to mitigate risks. So with DOE, we can use it to identify risks in terms of bad performance, like bad that will lead to bad response. And you can then propose a solution, propose a counter strategy. Yeah. So, and with that, the knowledge is fully utilized.
Now, for someone new to design of experiments, what's really the first practical step they could take to start using it in their lab?
Yeah, this is again challenging because sometimes the simple is much more challenging than the complex. So the first step I would say really to start with simple screening designs to support identification of best performing conditions. For instance, again, let's take an example in chromatography where you would like to identify what is the best LC column you can have for your application. So you can then do a screening of LC columns while studying other critical factors such as the pH of mobile phase, organic solvent, etc. So usually trying to focus on parameters that will impact on selectivity first. So second, we need to define clearly the responses that you are monitoring. Yeah, so in chromatography, it could be the resolution between critical pairs. It could be the tailing factor. So you can even model this later. But starting with simple designs can be very interesting. And I think something very nice is to learn from the patterns. Why? Because we are doing experiments and we get data. We need to interpret that data. So shift mindset from testing to understanding, not just getting the data and say this is my best condition. Really like using the data that the DOE is providing you, then perhaps advancing later as soon this is more like like better implemented, let's say, with more confidence. No? So perhaps the scientists could move to applying optimization, yeah, designs and create, why not, prediction models like the crystal balls. Yeah, I like the crystal balls because they tell me what to do without doing the experiments itself. So yeah, there are simpler and more advanced types of DOE, like for both screening and optimization, and using DOE software can also greatly help. I would encourage to learn what lies behind the DOE, but like using software, they can really help, selecting the best type of DOE for your application. Yeah.
When you say predictive modeling for LC methods, what does that actually look like in practice? What parameters tend to matter most when optimizing an LC method? pH, gradient, column chemistry, or temperature?
So I think number one is let's do risk assessment. Yeah. So this will totally depend on the type of chromatographic mode we have, the complexity of the sample, and the intended use of the method you are developing, but also the stage of development you are like. You are doing screening of conditions or are you doing optimization of the method? Yeah. So we tend to study factors that impact more on selectivity usually during screening. Okay. And later we can refine the ranges for those factors by doing some optimization studies, plus focusing on identifying sources of variability as well. So for instance, to be practical, like for reversed phase LC, yeah, so we could screen then LC columns chemistry, organic solvents can be important. Depending on the type of samples, like if you have ionizable compounds, it can be very critical for you to screen also pH of mobile phases. You screen like oven temperature, yeah, which can impact on selectivity, especially if you work with ionizable compounds, but also efficiency. Something that I didn't mention during our discussion on design of experiment, and it's worth mentioning here, is that the higher the number of factors studied, the higher the number of experiments. Yeah. And we may then prioritize factors for screening and then building optimization designs to refine the method. And then we can study secondary factors that impact more on efficiency. Perhaps another example in ion pairing chromatography. Yeah, so besides screening LC columns, why not we screen now the type of ion pairing reagent? So its concentration can really impact on selectivity, oven temperature, etc. So again, so we go back to the importance of doing risk assessment, where we need to understand the type of samples we have, the potential impurities that might be present in that sample, the physical chemical properties of those compounds, because with this at hand, we can do technology assessment, like which kind of chromatographic mode should I select, or perhaps on another technique. No? And then finally, we can define the best critical parameters to be studied.
Amanda, can you walk us through a specific example where AQbD saved time or maybe helped with a difficult
Yeah,
separation you were working on?
perhaps I can pick a method for impurities profiling of nucleic acid based therapeutics. This is also a hot topic, like very promising therapeutics right now, and they are very complex. Yeah, so we still need to find solutions for many things, and develop like orthogonal methods to evaluate their quality. So like using then chromatography is one of the golden standard techniques to analyze impurities of oligonucleotides. And very often people use like ion pairing chromatography. So we wanted to develop a method using ion pairing chromatography. And one of the challenge things we have there is that the impurities, they are like structure related impurities, very similar to the API. Yeah. And even changing like gradient, LC columns, different oven temperature and all that, it's quite challenging to separate those from the main API, no? The main ingredient, so that we can then quantify the impurities using chromatography. So then we have been applying design of experiments and the AQbD principles so that we can have a systematic approach where we define the experiment so that we can identify, for instance, the best LC columns that can provide the best separation between these impurities and the API. Also, the selection of the ion pairing reagents. So often there with oligonucleotides, we work with alkylamines. Okay. There are different type of alkylamines. Some of them, they are more hydrophobic. Some of them will be less hydrophobic. And this can greatly impact on the chromatographic separation mechanisms, which are, being favored. If you have more hydrophobic or less hydrophobic, this might, favor more or less ionic exchange chromatography or hydrophobic interactions. So it's really complex what is happening behind the ion pairing chromatography. And we decided to use design of experiments to screen the columns. And we even use an interesting tool, which is called desirability function, where we had several impurities. We wanted to improve the separation. Yeah. And we wanted to look to all those responses at the same time. So what can you do with that? Because I don't want to improve only the resolution of impurity 1 and 2. I want to improve the resolution between all the critical pairs I have, yeah, especially those very close to my API, my main ingredient. Yeah. So we use then desirability, which will calculate a desirability value, let's say, for each of the responses. Yeah. And then we can calculate a global desirability. And we can generate like very nice graphs showing us which column and which kind of ion pairing reagents, or even like we were screening oven temperature, which has a good impact on performance there as well. And also gradient slope, gradient program can also have a very great impact on performance. So we could, identify workable regions and the best columns so that we could pick really the best, so that we could have like good resolution between all the impurities, and then use LCUV to quantify impurities. You see that very often for oligonucleotides, because it's so challenging to separate impurities from the API, that often people, they use LCMS, because then you can distinguish the impurities by the mass. Even though they are, colluding with the API, you can quantify them using LCMS. So using a simple LCUV method is quite interesting for quality control, for instance. So it was really nice to show the power of using design of experiment and AQbD to select an optimal condition using a simple technique, LCUV.
Partially because I know that over the course of your career, you've worked in so many diverse areas. I was really interested to hear, what do you recall as the most challenging sample matrix that
Okay.
you've worked with?
Recently, I must say that one of the most challenging areas for me has been working with large molecules in chromatography, because their behavior is much more complex compared to small molecules. So you often deal with slow kinetics. So large molecules, they are lazy molecules, they move very slowly. So which can then lead to significant peak broadening and loss of sensitivity. And the method becomes much more sensitive to system related factors. So even small differences like, different dwell volumes, even when you change tubing size, which is connecting autosampler to the UV detector and all that, changing system dispersion can noticeably impact performance. That's terrible for transferring the methods. On top of that, I think like non-specific adsorption is a major challenge as well. like especially for metal sensitive compounds. And I can go back to oligonucleotides, which is an example. So oligonucleotides, they have like a negatively charged backbone because they have like phosphorodiester or phosphorothioester groups, yeah, which will be negatively charged in solution. And if they see a metal, they will just, love it. So interactions with metal surfaces in the LC flow path or column hardware can really impact recovery, reproducibility, sensitivity for those metal sensitive compounds. Yeah. Then scientists, they have been working with oligonucleotides and RNA therapeutics. They need to find actually solutions to mitigate these issues. Yeah. Another common type of non-specific adsorption is the ionic interaction. For instance, to analyze peptides. So we know that now peptides, some of them might have many amino acid residues, which are basic residues. Yeah. And then in solution, this might be positively charged. And then you're using reversed phase chromatography, you use a column in which we'll have, a higher percentage of silanol, residual silanol groups, which is negatively charged. Negatively charged, positively charged, they love each other, right? Which can then significantly affect the efficiency of peptides analysis. Yeah. So fortunately, this challenge, they have driven innovation in column technology, LC systems, all lab consumables. Yeah, so examples could include like low adsorption LC hardware, sec columns with hydrophilic coating, right, to decrease carryover, or LC columns with charged surface modifications, hybrid silica, and other type of technologies. It's really nice to see how all these complexities, these challenges that we face, how this is actually driving innovation in this space, in chromatography, in general.
What software or modeling tools do you rely on the most? And I'm sure it varies from different sample matrices, but what tools do you find to
Yeah,
be most
so in
helpful?
fact, it might not vary across the different type of samples. However, yes, depending on the type of technique that you are applying this, the AQbD principles, you might want to work with different kind of software. So for instance, for design of experiments, there are many excellent software options, including like Design Expert, but standard statistical software like Minitab, Jump, Mode, or other type of software as well. Some of these tools can also be even so you can work automatically. What does it mean? What do I mean by this? That the software can communicate directly with the chromatographic data system. Okay, driving actually creating all the instrument methods, creating the design for you, and then you can just go to the CDS, to the chromatographic data system platform, and you can hit start, and then the experiments will start automatically. Yeah, so for sure you prepare your samples and all. One example is like Fusion software. This can help streamline data transfer and analysis for the industry, is quite interesting. For retention modeling and method optimization, tools like DryLab, ACD Autochrome software, ChromSwar, they are very powerful. But again, this is more limiting to chromatography. Yeah. And they allow you to simulate chromatographic behavior under different conditions and also identify optimal method settings efficiently. Yeah? So, in addition, data analysis tool for trending, like monitoring of performance, can be very interesting. So, I know that we didn't expand our discussion to the procedure lifecycle management. Okay, the procedure lifecycle management is actually driven by analytical quality by design. And there are other stages of developing a method that can help build the quality in that method. And this is like late stages when you have the final method and you keep monitoring the performance. You can do this with control charts and all that. And then like using statistical software can also help, do this late stage activities, further improving the method.
And what skills beyond the instrument, whether it's statistics, coding, modeling, have helped you the most throughout your
Well,
career?
I think first getting a strong foundation in understanding the chromatographic fundamentals is important, as I discussed earlier. Like not just applying DOE will help you enhance knowledge. So you need to make sense of the data. And for that, it's important that you have a good and solid foundation in chromatography. I think this is the number one for me. But I won't lie that learning a little bit of statistics and chemometrics help it, you know. These have been essential for implementing design of experiments and identifying the best DOE types for the purposes I had, you know, and implementing AQbD principles, enabling efficient experiment design and meaningful interpretation of very complex data. I think this helped a lot. So these skills, they are also critical for validating predictive modeling. When you develop a model, you need to trust what the model is telling you. So then using statistical tools, you can, you know, validate the models and select the right tools for method validation as well. Yeah. So more broadly, they have given me a deeper understanding of the analytical procedure life cycle and the practical application of AQbD. And these definitely are two areas that have significantly advanced my career over the past few years, David.
Totally. No, that makes a lot of sense. And I know this is a very general question, but feel free to answer it however you would like. But what excites you most about the future of chromatography and
Well, there
separation
are so many
science in
things to
general?
highlight, but what excites me the most about the future of chromatography, I think, is the integration of modeling and artificial intelligence into method development. So these tools, they are helping us to gain deeper understanding of separation mechanisms and also shifting the field to predictive, knowledge driven approaches. This can especially help design solutions for the separation of more complex therapeutics. We discussed about some of the challenges that those complex therapeutics they bring, and people, they are just struggling to find solutions and innovate. So I'm also excited to see then the advances in technology that will be addressing this unique challenge that we discussed. And I also believe these innovations will really elevate the role, you know, of analytical science in general, making it even more central to decision making and quality assurance in the development and manufacture of medicines, because this is the area that I've been applying chromatography. So I'm quite excited in this sense. Yeah.
That's a great answer. And yeah, I knew that in terms of discussing the future, AI is always bound to be mentioned as part of that. But this has been great, Amanda. Was there anything that you wanted to talk about today that we
Well,
didn't
there
get
are so
into?
many challenges that we can share in chromatography, right? I'm really happy with the invite, David. In fact, yeah, it's nice to see the work you have been doing and sharing, you know, a little bit of the knowledge that all many great scientists they have across the globe. So it's really nice to see the work you have been doing, and it was really a pleasure to be part of it and share a little bit of what I've been doing. Yeah,
Absolutely.
thank
Thanks so
you.
much.